I'm Rick from BioVerge, and we're here with Reason and Morad of Repare Biotechnologies. As part of our diligence review, we're going to have Reason and Morad work through an investor presentation as well as a Q&A session. So I'll kick it over to Reason.
Okay, thank you very much. So Repare Biotechnologies, remember the top word here, repair. This is what we do. This is an important topic. What are we? So we're a preclinical biotech company. We're in the longevity industry, which is a very interesting new industry, which a lot of people are very interested in because doing something about aging is very important and also a very lucrative area of business in biotech in general. We are working on a way to reverse atherosclerosis and other hypercholesterolemias by providing cells a novel way to degrade excess and modified cholesterol. This is a first-in-class approach. Nobody else is doing this, and the existing standard of care is terrible for this type of disease. They really do no more than really pause disease progression. We believe we'll be the first to effectively reverse it using our approach. If everything goes great, we will be doing human clinical testing in 2022. We are, for the investors in the audience, a fairly standard issue, go through the FDA biotech company, so expect a timeline of 3 to 5 years to get to a liquidity event, which would be somewhere around phase 2 and either an acquisition or going public in order to raise funds for phase 3, etc. So I'm going to go rather fast with these next pieces because most of you are probably familiar with this, which is just to say that aging is an enormous market. Everybody gets it. Everybody needs therapies. It has an enormous cost, and anybody who can do things about it will make a great deal of money. The first groups that are doing things about it are indeed enormously valued. And I should say that the Life Biosciences is a little out of date here because I believe they're raising $100 million right now. What do you do about aging? Aging is a natural outgrowth of metabolism. It's damage that results in the same way as rust results in a metal structure or the same way that wear results in your car. It's natural, it happens. Don't try to mess around with metabolism to stop it happening. That's a fool's game. What you do is you find the damage when it's there in small amounts, and then you repair it. And that prevents it from causing age-related diseases. And we're just getting to the point in medical science where we can do this. So our goal is to do this for as many things as we can do this for. We know collectively a great many people in the aging community. We know a great deal of the work that is out there. I've been in this field for 15, 20 years, something like that, as a patient advocate, watching things come and go. So we identify what works. We go license the IP, bring in the inventors, do proof of concept, and then we go into clinical development with it. This is the plan. And what I'm going to show you today is one of our programs. We have one other at a state worth talking about, which we can talk about another time. But this is one. We're working on them first. We're getting it somewhere interesting. Atherosclerosis. Why atherosclerosis? Because it kills between 10 to 25% of everybody. Everybody. Quite a sizable fraction of the people watching this video, unless something is done, will die from this condition. If you're over the age of 40, this process of gunk building up in your arteries is already started. And you can actually see the lesions if you go do suitable scanning technologies. Not fun to think about. The reason this happens, and it's important to understand why, is that macrophage cells, which are a type of immune cell, normally work to keep your arteries clear. They get rid of this gunk. This works great when you're young. But when you're old, you have a bunch of oxidized cholesterol rather than normal cholesterol in your body. And these cells didn't evolve to survive that. They can't deal with it. They go crazy, they become inflammatory, they die. And a plaque is in fact basically a macrophage graveyard that calls in more macrophages all the time, and they die and it grows. And this will kill you eventually. It will destroy one of your blood vessels in such a way that you have a stroke or a heart attack and you die. Present standard of care is terrible. There is great unmet need. These numbers on the bottom left, 14 to 30% type of reduction of incidence, this is pathetic. This is a nothing. This is not, you're going to die from this. They're just moving the needle a little bit off a few years to the right, let you die of cancer first. And the modern generation, which is things that are super statins that can really reduce your cholesterol down to 10% or less of human normal. And frankly, it's interesting that people seem to be quite okay with that. But it doesn't really solve the problem. It doesn't reverse the plaques to a large enough degree. They still grow, you still die. So what we need to do is what we're doing. Treat it like damage and repair it. Don't try to slow it down. Actually go in there and get rid of it. So how we do that is we have licensed a set of degrading enzymes. These are proteins that break down cholesterol, which human cells do not normally do at all. So what we do is we provide cells in and around the plaque with these proteins via some form of gene therapy. And at that point, they cheerfully chew up all the cholesterol and get rid of it, whether it's oxidized or not. And you can see a paper reference there by the inventors of the technology. It's a bit of a dry read, but it's very interesting. So we have actually taken this from the university that developed it, and we've demonstrated that in actual fact, yes, it is great. This shows two sets of macrophage cells in a cell culture. What's happening here is we're feeding them oxidized cholesterol, and the one on the left doesn't have our proteins, and the one on the right does have our proteins. The green there is excess cholesterol. It's cells that have become completely incompetent and they can't hand off this cholesterol or get rid of it or deal with it in any way, shape, or form. They're pathological. There's a lot of them on the left. There are very, very few of them on the right because we have given these cells the ability to express our cholesterol degrading proteins. So they got rid of the cholesterol and they're happy and doing their business. This is in miniature what we want to do in the body. Now, if we look at what we can do in the body, this is using adeno-associated virus, AAV, which is a very good prototyping vector, but not something you want to take forward to the clinic because it's too expensive, but it just works. It's great if you're doing it as in mice. We provided a very high dose of these enzymes to mice. And as you can see, we've basically halved the cholesterol in their livers. We just injected it into a vein, off it goes, most of it goes to the liver if you do that. And so here we can see a very large effect after 2 weeks. And in the bloodstream, testing for cholesterol will actually also pick up some of the downstream metabolites produced by our degradation of cholesterol. So all of the cholesterol got chewed up in the liver, spit into the bloodstream, and there it is. This is actually quite safe. The mice in question, between the control mice and the treated mice, no difference in body weight, no difference in liver weight, and the normal detection of liver problems by AST, ALT enzyme ratios, which you can go to get blood tests for that yourself if you're interested, didn't have any great significant alterations. So there's no serious side effects produced by this therapy, which is great because that's really what we are after. We're after a safe basis for treatment. In terms of our IP, we are developing a patent that we licensed from the University of South Alabama, which covers the basics of this technology. And we ourselves, following the study results that I just showed you, filed a patent ourselves based on the unexpected behavior there. It's unexpected because the mice didn't die. If you went to anybody in the last few years and said, hey, I'm going to put an enzyme into the body that degrades cholesterol, they would have looked at you and nodded wisely and said, have fun with that. Tell us how it kills the mice. But it doesn't. It's actually very safe. What we're going to do with this, we're going to take this into HoFH, which is homozygous familial hypercholesterolemia, which is a rare disease. And it has the benefit that it's essentially accelerated atherosclerosis. These poor individuals have such a huge amount of cholesterol in their bloodstream because of their genetic aberration that they will die at age 30 from strokes and heart attacks because they have the arteries at that point of a 70-year-old because of the accelerated development of atherosclerosis. And they're often identified far too late. Can have significant lesions at the time in which they're identified. So it's a great market to come in and prove out a technology for treating atherosclerosis. And immediately upon getting that, we will then go for atherosclerosis. And there are numerous other indications. There's too many to list here, in which some part of your body, for some weird reason, has too much cholesterol. So we can go in and deal with that. Some of these are genetic rare conditions. Some of them are like fatty liver disease is a case where too many people are eating too much, but they're nonetheless, they're still treatable. Now, in terms of where we are and where we're heading, we expect to have a pre-IND meeting next year at some point with the FDA once we're far enough along in settling the formulation of our therapy and have our optimization done, and we've developed the animal models and completed them to show that this has efficacy. And then pretty much in concurrency with preparing for our pre-IND meeting, we'll be raising a Series A, or we'll be doing a Reg A plus raise, or we'll some other form of raising a very significant amount of money at a significantly higher valuation than we are now. So this is in the future. In terms of who we are, we have myself, I'm Bill. Both of us have been around in this community for a while. We've both been investors. We know a lot of people, and we're quite familiar with the scientific community and the business community. Morad is someone who's connected to us through the inventor of our technology here, who has a background with Pfizer and Harvard and really knows his way around preclinical development and clinical development for hypercholesterolemias. Bill Gannon keeps us on the straight and narrow. He's a one-man shop for talking to the FDA. He has a tremendous network of people who can set up the IND process very, very quickly. And Mark is our very talented principal scientist who manages our lab up here in upstate New York and keeps a handle on the fun end of the business. Our advisors, Richard Honkanen is the inventor of this technology. Sampath and Bobby are both specialists in the atherosclerosis, Sampath on the science side and Bobby on the business side. They're quite familiar with FDA process, and they've collaborated on multiple companies putting through multiple indications. David and Graham help us on respectively preclinical development and immunology. They're both rather more focused on one of our other programs, but they certainly have a use in this one. And Aubrey, of course, is Aubrey. We've known each other for donkey's years, and I'd have trouble keeping him off any of the boards of the companies that I would be on. So he actually introduced us to Richard Honkanen originally back in 2018. And that in summary is in a nutshell. We're quite happy to take questions about what we do, and I'm sure there will be a few coming up right now, in fact. Thank you for listening.
Thank you for that, Reason. So we do have some questions that we'll send your way, and others through the BioVerge platform might have follow-ons that we can facilitate later. But just to walk through some of those that we have on deck, it'd be great if you could walk through, do you mind stopping the screen share?
Sure.
There we go. So do you mind just walking through how you went about assembling the founding team as well as those advisors that you brought on?
Well, certainly. As I said, Bill and I sort of met because the investor community at the time, let's say 2016 or so, it was really small. There were very few people interested in angel investing in longevity-related companies back then, and indeed there weren't very many longevity-related companies, so we all ended up knowing each other. And then we were interested in doing something that was more than just putting money into the industry, and something turned out to be Repare Biotechnologies at the end of the day. Morad, we met via Richard Honkanen. Those two have known each other for some time, and so it was an obvious choice to bring him on because he already knows a lot about the program specifically that we're working on through his friendship with Richard, and his background kind of speaks for itself. He has a very impressive resume.
Appreciate that. For those meeting Repare for the first time, do you mind sharing more about the company's long-term vision?
Okay, the long-term vision here is really, as I said very early, skipped over very early in that presentation, is we're very big on the SENS view of aging, and that aging is basically a set of different forms of damage that then lead to pathology. We want to attack every one of those and produce meaningful therapies in as many are not really being done already. So for example, about the only one right now that I wouldn't go tackle would be Senolytics, to destroy senescent cells, since the 15 companies doing something there seem to have it well in hand. But there's many other areas where we really need to make some progress here. And there's an enormous opportunity for anyone who gets in there early to make progress. So there are any number of potential programs out there that look very much like our atherosclerosis program, where there's a first-in-class approach to repair some form of damage that will meaningfully change the whole playing field for everybody who suffers the condition and everybody who works in the field. And for some reason, nobody's trying it yet. So it just needs to be done. And I think perhaps it's that very few people have the sufficient breadth of reading to see more than one of these. So we would love to be the company that goes out there and does a bunch of this, and ends up looking like a Juvenescence, except bootstrapped from the ground rather than starting with $300 million.
So in pursuit of that vision, what do you see as the biggest challenges?
I think honestly, the biggest challenges right now is get the first program done. So we have to show ourselves to be competent in a program before we can really start diversifying enormously. So getting the cholesterol catabolism program to the clinic, and that means essentially right now putting all of the pieces together in our formulation. So we have to think about targeting, about efficiency, about what delivery mode we're choosing to use, and then the economics of manufacture all feed into that. So you're juggling 15 variables at once to come to a final thing. It's not just, is it working? And it's not even just, is it safe? Even though, frankly, in the modern environment, is it safe is much more important than is it working, at least from the FDA's point of view.
Great. What would you say is Repair's next closest milestone? And why will that have an impact on the company's ability to advance the project and fundraise at better terms in the near future?
So Morad, do you want to take that one, or shall I? I guess I will take that. So as I mentioned, we have to do this business of getting the formulation together. But the pre-step there is we have to show efficacy in animal models for all of these things. So we have to demonstrate that, yes, we actually have all the understanding of how exactly these proteins work in vitro and in vivo. We have to prove their function. We have to demonstrate that we understand how they work over time, what the kinetics are, and then we have to put them in animals and answer all the same questions there. And this is an ongoing process right now. And what this gives us, beyond the ability to say at the end of the day, okay, this is how we're going to deliver it. What this gives us at the end of the day is the ability to go to a big pharma entity or a major biotech investor and say, here, look at this package. We know how this works. It works. Let's talk turkey. So it opens doors for us, in short.
Let's talk dirty. That's great. Morad, did you have anything that you wanted to add? I think you might have had.
Sorry about that. I had issues with the connection. No, in fact, I think Reason sums it up pretty well. We really have to bolster our in vitro data together with our in vivo data so that we could present a compelling story to big pharma and we could partner with them. We have no doubt that we will hit our milestones and reach our goals. It's just that we have to get that done over the next 3 to 6 months.
That's great.
Indeed.
Thank you. So I think it'd be helpful if you could briefly describe Repair's competitive landscape, and specifically, how the repair therapy will be used to outright replace or be used in conjunction with the standard of care, such as statins.
Go for it, Morad.
So statins or PCSK9 inhibitors, what they basically do is they either inhibit the production of cholesterol in terms of statins, the endogenous cholesterol that we produce by inhibiting the key enzymes in the synthesis of cholesterol. And PCSK9 inhibitors, what they do is they actually upregulate the receptors that take away this bad cholesterol, the LDL cholesterol, away from the tissues. our approach is very, very different than those preceding technologies. Our approach is to actually degrade cholesterol and get rid of it from the body. So instead of just slowing down the process of atherosclerosis, what we aim to do is we aim to a first-in-class approach which goes and degrades cholesterol at target sites, which is in our case for atherosclerosis the medial layer of arteries. So it's very, very different. I don't think that we would be giving a disruptive technology. And in fact, I think that other companies would be more interested in partnering with us to actually further this technology rather than relying on statins and PCSK9 in the future.
Okay, last question. If Repare is successful in its mission, how will that impact patient lives?
Well, simply speaking, a lot of people will be alive who would otherwise be dead. But more than that, it's not just the fact that suddenly you drop dead from atherosclerosis, though it is a condition that is pretty notorious for that. In fact, that you think you're fine and then you're dead. Less of that. So, in fact, none of that we would hope. But at the end of the day, also, it's a condition that can cause very progressive, horrible loss of capacity towards heart failure because crucial veins and arteries are like narrowed, taking blood to where it needs to be. So you'll find people getting stents these days, for example, to widen their arteries. And that has a very strong, it makes a very big difference to your quality of life, which is the only reason people would undergo horribly invasive processes like that. It changes your energy, your ability to do things. People can climb stairs where they couldn't before, that sort of thing. And we would anticipate that if you can clear out gunk from your arteries, then stents might go the way of the dinosaur in an ideal world. They're not the best solution in comparison to getting rid of the stuff that's actually narrowing your arteries in the first place. Any other thoughts on that, Morad?
Well, yes, and we could also make the analogy to cancer therapy. Current cancer therapy is aimed at making all patients cancer-free. Right? The current sort of standard of care for atherosclerosis is let's sort of reduce your LDL levels to a certain point where we don't have any more progression of this ticking time bomb that's still in your arteries. So it should be unacceptable in the future to actually accept when your physician tells you, well, you know what, we've slowed down the progression of atherosclerosis, but we haven't gotten rid of it. It's like saying we've slowed down the progression of your cancer, but we haven't gotten rid of it. That would be unacceptable at this time. And I hope that in the future, it would be unacceptable for patients to go around or people to walk around with atherosclerotic plaques.
Absolutely.
That is a beautiful vision. Well, that's all we got. I want to thank you both for joining us and taking the time to go through what you're working on. We couldn't be more excited about that and the opportunity to work with you. And we hope that those that are meeting you for the first time today are just as excited. For those watching, if you have any questions that weren't answered in the presentation, please feel free to pass those along to us and we'll make sure that they do get answered. You can reach us by email at info@bioverge.com and want to thank you as well for tuning in. So I hope everyone has a great day and catch you next time.